Hello, I'm Lauren Crew, and I work in the Hyman in vivo electrophysiology lab here at UNLV, where our aim is to understand how brain area interactions contribute to cognitive processes such as learning and memory in psychiatric disorders such as schizophrenia. Schizophrenia is a mental disorder characterized by continuous or relapsing episodes of psychosis that have positive and negative symptoms. Some positive symptoms include hallucinations, like hearing voices or seeing things, delusions, paranoia, and disorganized thinking. Some negative symptoms include social withdrawal, decreased emotional expression like depression, and apathy. Communication between two areas of the brain, the hippocampus and anterior cingulate cortex, which is a prominent structure located in the prefrontal cortex, is known to contribute to cognitive function and is seen to be disrupted in patients with schizophrenia. Many genes have been associated with the development of schizophrenia. However, the underlying genetic processes that result in the symptoms seen in schizophrenia has not been well studied. With the ability to collaborate with the neurogenetics lab here at UNLV, we are able to dive in deeper into the possible mechanisms genes have on schizophrenia. One known isoform, or isolated form of a gene, linked to schizophrenia is PCM1. PCM1 has shown to affect neurogenesis, primary cilia functioning, and actually leads to behavioral phenotypes related to schizophrenia. Primary cilium acts as an antenna on a cell that surveys the extracellular environment, where they receive various signals and transmit those signals into the cell. So we hypothesize that dysregulated cilia functioning is what leads to disruptions in network oscillatory activity between the ACC and hippocampus seen in schizophrenia. So to investigate this, we created a genetic mutated mouse without the PCM1 isoform, or a PCM1 knockout, and implanted them with 8 wires aimed at the hippocampus and the ACC, and then tested their behavior using a novel object recognition task. In this task, animals are presented with 2 novel objects to explore. After 10 minutes of exploration, animals are placed back into the environment with one of the previous familiar objects and a new novel object. The idea is that animals will explore the novel object more frequently if they remember the familiar object. However, we actually found no significant differences in behavior during the novel object recognition task. This may be related in part to a low N, but in reality, we found that this is not a good measure of cognitive performance, as mice generally don't explore the novel objects generally. They tend to veer towards the sides of the environment than actually exploring the objects in the center. They may be scared or uninterested. Either way, I did another analysis recently that does show significant differences in areas explored and distance traveled, where PCM1 knockouts generally explored less than healthy animals, so suggesting that there is some form of cognitive impairment in these PCM1 animals. So this is great, but what's going on in the brain? Well, under normal circumstances of thought, we usually have ensembles of neurons that activate and deactivate together. This oscillatory pattern is frequently seen between 4 to 12 hertz, which is commonly known as the theta rhythm, which you can actually see as the bright yellow thick line in figure 4. However, as you can see in the PCM1 knockouts, we didn't see that. Instead, we hardly see any theta, and actually some instances of lower frequency oscillations, which is what we similarly see in patients with schizophrenia. As far as communication between areas goes, in healthy animals, we see that hippocampus theta is synchronized to ACC theta. So they're communicating with each other. These theta oscillations are occurring together in the same period of time, which is actually required for appropriate cognitive functioning. But in PCM1 knockouts, we don't see that nice, thick, solid line, indicating that these areas are not communicating. This supports the desynchrony hypothesis of schizophrenia, which suggests that the symptoms seen in schizophrenia are a result of a lack of communication between brain areas. Finally, what isn't shown here, because it was done by our collaborators, is that PCM1 also caused dysfunctionality at the primary cilia, thus rejecting our null hypothesis and suggesting that this disruption in brain network activity is likely due to improper primary cilia functioning. This leads to the idea that schizophrenia may be a result of underlying biological processes that ultimately just needs to be further investigated. Thank you.